Hi everyone, and welcome to the last recorded lecture for Anatomy and Physiology 1 on sense organs. So this is chapter 16, and we'll talk about the sensory system and some of the sense organs. So our senses inform us of changes in our internal and external environments. Right? We keep on bringing up the idea of that the brain is enclosed in a skull. It's pretty dark in there. So how does the brain know what's going on? Right? We need to have sensory receptors. We have many different kinds of sensory receptors throughout our body, both external and internal. And these are used to detect changes in the environment. And they then send these nerve impulses to the brain, which can then initiate a response. So that's the idea of senses. They're going to inform us of any changes inside our body or outside of our body. So our senses are our way to know what's going on. And senses can be divided into general senses and special senses. So the general senses include pain, touch, temperature, feeling of pressure, balance, stretching, and also chemical changes like pH. So the general senses, these receptors are scattered throughout our body. For example, we have nociceptors that detect pain throughout our entire body, almost everywhere. The special senses are what you're familiar with when you talk about the sensory system. So taste, which is also known as the gustatory system, smell, or the olfactory system, vision, hearing, and equilibrium. Those are the special senses. So these are associated with a specific sense organ. And each sense depends mostly on the sensory receptor. So the sensory receptor is any structure that's specialized to detect a stimulus. So it detects a stimulus, it detects a change, and then it will form a nerve impulse in a sensory neuron. So a sensory neuron is a neuron that carries a nerve impulse to the CNS, right? We spoke about that in chapter 12, that sensory neurons are afferent. They carry nerve impulses to the CNS, including the brain. And the brain will then interpret that nerve impulse. And I'll talk about what a sense organ is in a bit, but a sense organ is any structure that combines nervous tissue with another type of tissue that tries to enhance the response to a certain type of stimulus. So a sensory receptor is any structure that could detect a stimulus, and if it has extra types of tissue, then we call that a sense organ, not just a sensory receptor. Remember, an organ is two or more types of tissue that work together to perform the same function. Right, we spoke about that in chapter 1. So let's talk about some of the parts of stimulus transduction. Each type of receptor can respond to a specific type of stimulus. And the nerve impulse from the receptor, from the sensory receptor, will then go to the central nervous system by spinal nerves or cranial nerves. So the sensory receptor is in the periphery. Then those sensory receptors branch into either cranial nerves or spinal nerves, and then those feed into the central nervous system. And the final destination is always the brain. That's the always final destination. So the first part is where the sensory receptor converts the stimulus energy into nerve signals. So the stimulus can be, let's say, a pattern of light. It can be heat. It could be sound waves. Those are stimuli, right? So it could be a sound or a smell. That's a stimulus. And that can activate a sensory receptor. And that sensory receptor will convert the stimulus energy into nerve signals. That process is called transduction. That whole process is called transduction. Transduction just means the conversion of one form of energy into another. So what we're doing in the sensory system is the sensory receptors are transducing the stimulus energy. Let's say it's a sound or a smell, that's a form of energy. And the receptor is converting that energy, is transducing that energy into nerve signals. And those nerve signals could then inform the brain of what that stimulus is. So the first part is there's a stimulus. So let's say this coffee smell, the smell of coffee. The first thing that's going to happen is it'll interact with our nasal epithelium. So sensory receptors in our nose will then pick up this stimulus. So the aroma, the molecules are the stimulus in this case, the molecules of odorant from the coffee, then interact with the receptors in the nose. That is called transduction because you have molecules in the air, that's one form of energy, convert into another form of energy. They interact with the nasal epithelium, which generate action potentials. Those are nerve signals, right? So an odorant or a smell molecule is transduced. It's converted into an action potential. And if that message, if that stimulus gets sent to the CNS, the central nervous system, we call that a sensation. So not all sensory signals go to the brain, but when they do, we may experience a sensation. And a sensation is a subjective awareness of a stimulus. So many sensations are filtered out by the brainstem. And a sensation is usually conscious. It's usually a subjective conscious awareness of something. So it's when you actually smell the coffee, that's the sensation. But right now, in fact, if you have a coffee machine, there are many other smells around you. You might not be aware of them. You might not be sensing them. But sure enough, there is the smell of other things in your house. But when the coffee machine is ready, that's the predominant smell. So that's what you're sensing the most. And the sensation created is determined by the brain area receiving the nerve impulses. So with the olfactory sense, we're not going to have the olfactory epithelium linked to the occipital lobe, for example, right? Because the occipital lobe is responsible for visual processing. So the olfactory epithelium has to go through the cranial nerve, the appropriate cranial nerve, the olfactory nerve 1, which then has to go to the olfactory cortex and parts of the temporal lobe. So it depends. The sense is rooted to a specific part of the brain, and the brain will then process those nerve signals and interpret it as a sensation. So the sensation created is determined by the brain area receiving the nerve impulse. In the case of a light stimulus, so let's say I'm visualizing this butterfly, and the way that the light is bouncing off of this butterfly is hitting my retina in the back of my eye. So there's a pattern of action potentials that are generated when I see this butterfly. And those action potentials, right, were transduced. The light energy that was bounced off of the butterfly hit my retina, and then my cells of the retina are transducing the energy from light into neural signals. And if my occipital lobe gets that information, it'll be sensing it. And then once that information gets to the cerebral cortex, then I can perceive it. That's what we call a perception. So the conscious awareness or the perception results from nerve impulses reaching the cerebral cortex. So a sensation is an awareness of a stimulus, and we really call it perception once it reaches the cerebral cortex. The book doesn't go specifically into the definition of perception. So right now we could use sensation and perception kind of interchangeably. The way that a lot of books explain it is that a sensation is something that even if like your gut detects something, that's still a sensation, even though you're not aware of it in the cerebral cortex. That's just perception of it. So it's a little confusing, sensation versus perception. Perception is always the conscious awareness of a sensation. So again, stimulus gets converted into action potentials, which are then sensed either consciously or unconsciously. When they're sensed consciously, we call that a perception. So sensory receptors transmit 4 different kinds of information. Sorry, you can hear my cat in the background, which is being very annoying. So I apologize about that. So sensory receptors transmit 4 different kinds of information. There's modality, location, intensity, and duration. And you do not have to memorize these as 4 things. I just want you to understand the concepts. Modality refers to the type of stimulus that a sense receptor receives. The type of stimulus that a sense produces. So what this means is the modality could be the temperature modality, which means this is a type of receptor that can produce a signal in response to temperature. So it's like, what kind of mode is it? If a sense receptor is in your retina, that means it responds to vision. The modality, right, is vision. So a hearing is another type of sensory modality. So it can be so that's the modality is all hearing. But then that receptor can either respond to, let's say, high frequency or low frequency in terms of that modality. You can also have a taste modality, and that can be a salty modality or a sweet modality. So it just means it's the type of stimulus that a sensory receptor produces. That's one way that different sense receptors differ. It's the type of stimulus or sensation that receptor can produce. The location is also encoded by which nerve fibers issue signals to the brain. So this means which nerve fibers are firing will determine what type of sensory experience you're going to get. So different nerve fibers can only be fired in certain receptive fields. So what that means is the receptive field is the area where a sensory neuron can detect a stimulus. It's like the sensitive area. So if a receptive field is very large, that means that a neuron can detect any stimulus within that one large area. But let me show you an example of, this is called the 2-point discrimination test. So here we have one sensory neuron. This is a myelinated neuron that's reaching the epidermis. So right here you can see the dermal papillae. So it's just in the epidermis. And you can also see that there are 2 points. And I want you to think about, how do you think, if you were pricked with 2 points, and this is one neuron over here, how would you perceive that? What would your brain tell you is happening if 2 points are hitting this one neuron? So this is one receptive field. And what that means is, regardless of how many points you put in here, your brain is still going to interpret this as just 1 point. Because this receptive field, it's like a yes or no answer. Is there a stimulus? And if this neuron is stimulated, you're going to feel a point there. Your brain is going to identify some kind of stimulation in this receptive field. And that's the case of most parts of our body. We have neurons that are pretty spread apart. So if like in the back of your shoulder, you wouldn't be able to discriminate between 1 or 2 points very easily because the receptive fields are quite large. If you compare that to like your fingers, so like on the tips of your fingers they're very, very sensitive. They're very ticklish because they have a lot of receptive fields there. The areas within which a sensory neuron can detect a stimulus is a lot smaller. So if you were to do this 2-point discrimination test, you would be easily able to discriminate between 2 points, because each point here would be hitting a different neuron in a different receptive field. So it's a lot easier to discriminate between 2 different stimuli when there are smaller receptive fields compared to when there's a large receptive field. So just for example, in the skin, one sensory neuron can cover an area as large as 7 centimeters in diameter. That's almost 3 inches. So no matter where the skin is touched within that 3-inch field, it stimulates the same neuron, and the brain is unable to tell if it's touched at the farthest end of the 3 inches or the other end. As long as you hit it anywhere within that receptive field, the brain will interpret it as 1 point. This is one of the lab experiments that we have as part of Lab 9 that we're not going to do. But this is a fun test to do with a partner or at home with a family member. If you take a paper clip, 2 paper clips, and you put them very close together, try to see how far apart you can go before you can tell that it's 2 separate points. So you start off very close together, and your brain is going to figure that out as 1 point. So you see how far apart the 2 paper clips have to go before your brain can say, oh, those are 2 separate points. And you'll see, well, I don't want to give away the answer, but you should make a hypothesis. Depending where you put the paper clips on your body, right, you might have different perceptions, right? You might have a different idea of how many points you're being pricked with, depending on where your body is. So if you did it in the back of your neck, think about how that would be different compared to if you did it on your fingers, your fingertips. Would you be able to pick up a smaller distance between the 2 paper clips on your neck or on your fingertips? So duration is also encoded by the sense receptors. So how long the stimulus lasts is encoded by the firing frequency within the passage of time. So basically what this means is how frequently the neurons fire and how long the stimulus lasts will be able to be transmitted by the sensory receptors. So let me give this as an example. The amount of time we hear a certain sound is encoded by the frequency through which certain neurons in our ear fire. If they stop firing, then we will stop receiving that perception of sound. So that's a kind of obvious one, right? So duration, right, how long a stimulus lasts, is encoded by how the firing frequency changes over time. And there's this idea of habituation or adaptation, that once a stimulus lasts a very long time, our neurons kind of get tired of firing and they stop. And that's when we get accustomed to something and it fades into the background. And I'll talk about this in a few slides from here. And finally, the intensity. So the intensity refers to whether a stimulus is very strong or weak. And intensity is encoded in 2 different ways. So the brain can distinguish the stimulus intensity by the number of neurons that are firing and the speed at which the neurons are firing. So the intensity is determined by the frequency of nerve impulse transduction by the receptor. The greater the stimulus intensity, the greater the frequency of receptor nerve impulses. So let's take sound as a modality. If it's a loud sound, that means that's a greater stimulus intensity. That means those receptors will be firing faster in your ear in response to a loud sound, in response to a quiet sound. For a touch, that's a different modality. If it's very, very hot, it's going to respond, your thermoreceptors are going to fire faster than if it's just warm. So again, some of these are kind of obvious, but receptors transmit all these kinds of information, right? Modality, which means the type of stimulus. Location, meaning where is it in the body. Intensity, like how strong is the stimulus. and duration. How long does the stimulus last? All four of these types of information are taken into account by sensory receptors. So like I mentioned before, an adaptation is a decline in the rate of nerve impulse formation due to repeated stimulation by the same stimulus. So when you first enter a room, you might smell a strong perfume, but if you wait a few minutes, or even 30 seconds sometimes, your brain adapts and you decline. The same rate of impulses is from the nose to the brain changes. It's decreasing over time because you get used to it. And this is good, right? Evolutionarily speaking. It's, okay, we're aware of a smell, but how much do I want that to affect me? I have better things to do than just focus on this one annoying smell. So my brain habituates, it adapts, and it says, okay, I'm going to ignore that for now. I'm going to move on to more important things. So a lot of the times we think about this, if you're living in the city and you hear a lot of taxi cabs usually, then you kind of drown out the noise of all the honking after a while. You habituate. Right, and this is preventing the nervous system from being overloaded by unimportant stimuli. An exception to this is with pain. So we do not become habituated to pain with pain receptors. So that's a common misconception, that you can adapt to pain. Some people have different thresholds for pain, but once you don't become habituated to pain. Once adaptation occurs, you need a stronger stimulus to gain a response. So if you become habituated to a certain smell of perfume, you would need to spray more of that perfume in order for another action potential to be generated by your olfactory epithelium. So now let's talk about different types of receptors. Receptors for the general senses are very simple. So the general senses include like touch and pain and vibration, stretch, all that stuff. So these consist of one or a few sensory nerve fibers, possibly with some connective tissue. So you can have a very simple receptor, which is as simple as just a sensory neuron with 3 nerve endings, and that can respond to the stimulus directly. Or you can have an enclosed, or what's called an encapsulated nerve ending, which is a nerve fiber wrapped in glial cells. In this case, right, if it's the central nervous system, it would be oligodendrocytes. If it's the peripheral nervous system, they're Schwann cells. And this is connective tissue that's wrapped around the nerve ending. And what this usually does is it enhances the sensitivity of the nerve fiber, or it makes it more selective with respect to which modality it can respond to. So these encapsulated nerve endings are usually more complex and more sensitive. So regardless, we have the cell body that's out of the way, right? In unipolar sensory neurons. So we have the nerve ending can be either free or encapsulated in connective tissue. The actual axon can be unmyelinated or myelinated. The cell body is always out of the way in a unipolar neuron. So we'll talk about different types of general sense receptors first, by the type of stimulus they detect. So a thermoreceptor detects temperature, right? They respond to heat and cold. We have thermoreceptors that respond to cold and thermoreceptors that respond to heat. We have free nerve endings in the deep dermis that detect temperature. And if it's above 45 degrees Celsius, then we feel burn. If it's below 10 degrees Celsius, we feel like frostbite. So that's the pain receptors in response to thermoreceptors. So there's a connection between thermoreceptors and nociceptors, which are pain receptors. And those can respond to changes in heat, like I just mentioned, but also to tissue damage and to certain chemicals that are toxic to the body. And we have free nerve endings everywhere in the body except for the brain. So that's why you can cut into the brain and not feel it. Right, so we said tissue damage and extreme hot and cold can stimulate nociceptors and causing the brain to detect pain. Next, we have proprioceptors, which detect body position. And these receptors sense the position and the movement of the body in real time. So as you adjust your position, your proprioceptors are aware of that. So we have those spindle cells, if you remember. We have receptors in our skeletal muscle and our tendons that know when the muscles contract, when they're relaxed, and in what position they're in. And these constantly inform our brain of where we are in space. Right, so the proprioceptors inform the brain about the positioning of the body and whether it's moving or whether it's stationary. Chemoreceptors are receptors that detect chemicals. So these are specialized neurons that monitor body fluids for chemical changes, like pH. So we have special receptors in our brain that detect any pH imbalances. Also, ion concentration. Do we have enough sodium? And do we have enough glucose in our blood? So these are all chemoreceptors that respond to chemicals. This also includes odors, right? Those are also chemicals. And these nerve impulses from the chemoreceptors are not processed by the cerebral cortex right away. Right? We're never aware of pH until the doctor tells us that we have a pH problem with our blood. Right? We're not aware of a lot of the chemoreceptors. So the nerve impulses from chemoreceptors in our body are not processed by the cerebral cortex. We're not consciously aware of blood glucose changes or sodium concentration changes. But some chemoreceptors in our nose, for example, and our tongue can be processed by the cerebral cortex if it's in terms of taste. But with the general, remember we're talking about general senses. So general chemoreceptors are not consciously perceived. Photoreceptors detect light. So light is made up of photons, and photons can stimulate action potentials. So these are different kinds of receptors. Mechanoreceptors are a large category of receptors that respond to any physical deformation of the cells or tissue. So if you touch something, if you displace tissue, right, if you deform any type of tissue, you're going to be stimulating mechanoreceptors. So these receptors, there's so many kinds of different mechanoreceptors that can detect pressure. There are others that detect vibration, some that detect stretch, some that detect very light touch versus very pointy, like pain, very sharp touch. So mechanoreceptors are the general class of receptors that respond to any physical deformation of the cells or tissues that are caused by vibration, touch, pressure, stretch, or tension. So here's an example of this sensory neuron that's in the epithelium. This is a touch receptor or a tactile receptor. So if this were in my fingertips, if I press on the epidermis, that's deforming the cells, right? When you're pressing on, you're applying pressure to these epithelial cells, that's applying some kind of disturbance, and that will activate the dendrites of this tactile cell, which will send the information down this axon to the peripheral nerves, the right spinal nerve, and then our brain will perceive that as a touch. We just felt something. We just felt some kind of a pressure. And of course, our brain tells us where this stimulus was detected because of projection. So once the cerebral cortex receives the information of the stimulus, the cerebral cortex projects the sensation back to the body region where that nerve impulse originated. So for example, when we press our finger down on the table, that message is getting sent to our brain, but where in the brain is it going? Right? We spoke about the somatosensory cortex. So the postcentral gyrus of the parietal lobe is going to receive a signal from our finger, right, in the specific location where our brain knows it's the finger. So we have a specific region of our parietal lobe that's responsible for our left finger, for example. So when we press our left finger down, that message is being sent to the specific location in our postcentral gyrus of our parietal lobe that's responsible for our left finger. And then when that part of the brain gets activated, it gets projected back to our left finger, so we feel it in our left finger. It's a little complicated, but this idea of projection, right, is the somatosensory cortex allows us to perceive where the sensation originates, and the cerebral cortex will project that sensation back to where it originated. So the pathways followed by sensory signals to the central nervous system, those are all called projection pathways. And most somatosensory signals go by 3 neurons. There's like a 3-neuron route to the brain. There's a first-order neuron, a second-order neuron, and a third-order neuron. I'm not going to go into too much details about this, but it becomes important when we talk about the touch pathway. And this is one thing I want you to understand. I want you to know the basic pathway. How does our brain perceive touch? What's the pathway from a nociceptor to the somatosensory cortex? So the best way to understand that is I really like this link over here. So I would highly recommend, well, you can't see that here. So I'm going to try to pause this 1 second. I'm going to pull up this link. And this is a really good link that talks about the pathway involved for the perception of touch, how you feel, how you know that you're touching something. And that's illustrated in this diagram from the book in terms of a nociceptor touching a tack. So I'm going to pause here. So here I am at the Get Body Smart website, and I just want to illustrate the 3 steps to the touch pathway. So this is showing how a receptor in the skin is going to signal to the brain that it's been touched, it's been contacted. And how does the brain project that back, that we are actually aware that our skin has been contacted? So the first step is we have a sensory neuron. This is our first order neuron. So the sensory receptor is the first order neuron that's in the skin, and in response to contact, so if I'm pressing down on the skin, that sensory receptor will be activated. And what we know about sensory receptors is that their cell body is far away. It's in the central nervous system, specifically in the dorsal root ganglion of the spinal cord. And that signal is going to be sent by this first order neuron through the dorsal column of the spinal cord. So depending where in the body the skin receptor is located, it's going to activate a specific spinal nerve. That spinal nerve will then go into the spinal cord. And in the dorsal column, that nerve will go up to the medulla. And in the medulla, specifically in the medial lemniscus, you don't have to know that term, but there is an interneuron in the brainstem that will then transmit this message to the thalamus. This is called our second order neuron. So the neuron that goes from the brainstem I want to see the whole thing. So that's the first step. right? That was the second step. So this is all of our first order neuron. So again, Sensorioceptor, ganglion, spinal cord, all the way up to the brainstem. Now we're ready for the second order neuron to take that from the brainstem to the thalamus. This is our second order neuron. Remember, the thalamus is then the relay station. It's going to interpret all these signals being sent, and then the thalamus says, okay, that's a touch signal. That's a touch signal from the lower part of the body. I'm going to send that to the somatosensory cortex and the part of the body that corresponds to this sensory receptor. So every part of the body has a specific location on the somatosensory cortex that it's connected to. So finally, we need our third order neuron to go from the thalamus to the somatosensory cortex so we can perceive the contact. So that's the three-step process. So first order neuron goes from sensorioreceptor all the way to the brainstem. The second order neuron goes from brainstem to thalamus. The third order neuron goes from thalamus to cerebral cortex. So I'd like you to know that general three-step pathway. And here you can see that's... So I'm not going to talk too much about the pain pathway here, actually, because there are two separate tracts. But again, the basic idea is there's a first order nerve fiber that could conduct a pain signal that goes to the spinal cord. Then from the spinal cord, you have a second order nerve fiber that goes from the spinal cord to the thalamus. And then you have a third order nerve fiber that goes from the thalamus to the somatosensory cortex, to the cerebral cortex. So that's a three-step process for pain detection. Your nociceptor feeds into the spinal cord to thalamus, thalamus, cerebral cortex. That's the pain pathway. So now we're going to move on to special senses that are all perceived by sense organs. So special senses are perceived by sense organs, meaning the eye, ear, nose, and tongue. So we do have some general sense receptors that are required for our special senses. So we said photoreceptors detect light. But we have photoreceptors in our retina, right in the back of our eye, that detect light for vision. But the eye is a sense organ because there's all these different kinds of tissues. You have blood, and you have fat, and you have connective tissues, and you have neurons. You have all these different kinds of cells in an eye, not just the photoreceptors. So it's a sense organ. And we call that, then vision is therefore a special sense because it's perceived by a sense organ. Chemoreceptors in the nose and tongue are responsible for smell and taste. So chemoreceptors detect chemicals in the nose for smell or chemicals on the tongue for taste. And again, since our tongue and nose have many different types of cells, not just sensory receptors, we call those sense organs. And therefore, we have taste and smell considered special senses. Believe it or not, mechanoreceptors in the ear are critical for hearing and equilibrium. So mechanoreceptors are activated by any type of displacement. So even sound is actually called an acoustic disturbance. So any change in the air frequency, any change will hit your eardrum and will activate certain receptors that respond to the change in air frequency. Also, we have mechanoreceptors in our ear that are responsible for equilibrium, so like knowing when we're properly balanced. So when we bend over, certain hair cells change position, and that causes displacement of certain sensory cells that tell our brain that we're changing our position, we're moving our head to the side. So that's caused by physical displacement of cells, and that's, again, what mechanoreceptors do. They respond to physical displacement and transduce that displacement into nerve signals. So now we will talk about the first special sense, smell. And a good way to start this is just by watching this crash course. The crash course covers taste and smell. Just a fun fact is we possess over 350 types of different olfactory receptors. So we have 350 different types of receptors that each can pick up one type of smell, one type of odorant. And we have thousands of different receptor genes, but many are called pseudogenes because they're not functional. So we have thousands of different olfactory receptor genes, but many of them can't really pick up any smell that we're aware of. They're kind of just fake. But we have 350 types that actually can pick up smell. And each of these receptors can only pick up one type of odorant. The average person can distinguish between 2,000 and 4,000 different odors. Some people can detect up to 10,000. And it's very hard to kind of group different odors because it depends how the brain processes them. So when we detect a smell, it's a combination of many different odorants. Recent research has indicated that women can detect a wider range of odors than men. And we now know that odor reception or odor detection decreases with increased age because of the hair cells get sensitized, or they get accustomed and tired, basically, of picking up all these senses that there's a loss of olfactory receptors, and they become desensitized. So as we age, we become less sensitive to taste and smell. And another fun fact is that olfactory receptors are the only neurons that are exposed to the outside. So your nostrils, right, in your epithelium, specifically in the mucosa, we have receptors, olfactory receptors in the mucosa of the nasal cavity that feed directly to the brain. So it's the only part of the nervous system that's directly exposed to the outside environment. Our olfactory receptors live for only about 60 days, so they're pretty short-lived, and they constantly get resynthesized. So now let's talk about some of the anatomy in smell. And let's start in the nose. So the mucosa, the olfactory mucosa, is the superior portion of the nasal cavity. And you can remind yourself, what is mucosa? What does it actually mean to be, what is a mucous membrane? Right? And we know that it's pseudostratified, ciliated, columnar epithelium, overlaying loose connective tissue. And this is exactly what this olfactory mucosa is. Right? So that's this question. So the mucosa is composed of olfactory receptors. So this is the mucosa. And here you have the receptors that are called olfactory receptors. And they themselves have special cilia. They have cilia. And they're surrounded by supporting epithelial cells. Right? And these are columnar cells. And of course, we have a mucus layer that helps trap any pathogens, but it's also responsible for making the odorants soluble. So it's important to make all the odorants soluble through the mucus so they can be detected by these cilia. Same idea as saliva in the tongue. You need saliva to help make the taste molecules soluble because otherwise you wouldn't be able to detect that. So we have the olfactory mucosa. Only part of the mucosa has these receptors, though. So not the entire mucosa does not have olfactory receptors. The olfactory receptors are only in some part of the nose. And again, we have these special receptors that have the cilia that are exposed to the particles in the nasal passage. So the olfactory receptors are also called olfactory sensory neurons because they're feeding directly to the central nervous system. This is the brain. This is the cranial nerve that feeds into the brain. So this is the direct route, this sensory neuron. Another thing to think about is what kinds of cells are these neurons? Are these unipolar, bipolar, or multipolar? So that's going to come up in a bit. So you should think about that. This is an interesting type of cell over here. So we have this is the mucosa. The mucosa, we said, has pseudostratified columnar epithelium that makes mucus. The mucus can trap any pathogens. And we have these cilia on the olfactory receptors that can be exposed to any odorants, any smell molecules in the nasal passage. So now let's talk about how the anatomy ties into the physiology and the actual sense of smell. So we said that chemicals that are dissolved in the air, so they're all odorants that are in the air already, they have to dissolve in the mucus layer first in order to stimulate these olfactory receptors. So they dissolve in the mucus, and the odor molecules each have to find a specific receptor on an olfactory hair, on these little cilia. So each receptor is only responsible for binding to one specific smell molecule. Once that smell molecule finds its receptor on the olfactory hair, then it could send a message down the olfactory receptor. And there are two types of odorant molecules that it could be either hydrophilic, which can go directly through the mucus and interact with the cilia, these olfactory cilia, or the hydrophobic ones that can't interact with the watery mucus have to bind to special proteins that help transport the odor molecule through the mucus so they can interact with the cilia of the olfactory receptors. So regardless of whether so we have odorant binding proteins that bind hydrophobic odorants. Hydrophilic odorants can diffuse freely through the mucus of the epithelium and bind directly to a receptor. So that's step one, right? The odor molecule binds to a membrane receptor on the olfactory hair. In response to an odorant binding to a receptor, on the inside of the cell, a G protein is activated, and the cyclic AMP second messenger system is activated inside the olfactory cell. This will lead to depolarization of this olfactory receptor, and that will depolarize the membrane and trigger an action potential in the olfactory nerves that travel to the brain. So this, let's take this cell over here. Here's an odor molecule. This one was hydrophilic, so it diffused through the mucus, and it just happened to interact with this receptor over here. This was one receptor that picks up the smell of vanilla. So this is a vanilla molecule. This is a vanilla odor olfactory receptor. So vanilla is a hydrophilic molecule. It diffuses through the mucus, interacts with this olfactory receptor, and in response to binding to that vanilla odorant receptor, it allows sodium inside, because a G protein was activated and cyclic AMP built up, and it opened sodium channels that allowed sodium inside this receptor that depolarizes it. And this could then cause an action potential in these sensory neurons of the olfactory bulb. So that's how we get a signal from the outside to the inside. So the cyclic AMP system is going to open up ion channels first. and that's going to cause depolarization. And those signals will then be sent to the brain. So when the olfactory fibers pass through, they have to pass through the roof of the nose. They enter a pair of olfactory bulbs. So this is called the olfactory bulb. And here is where they synapse with dendrites of other kinds of cells that feed in directly to the olfactory to the part of the brain that processes smell. So we have the cilia in the upper portion of the nasal passage, and then they have to go through the cribriform plate. We have to go through this the bony portion, through these little foramina, and that's where they interact with the olfactory bulb. And the olfactory bulb leads through the olfactory tract to the olfactory cortex. So the primary olfactory cortex is in the temporal lobe, and that's the final, or the almost final destination. So remember, the temporal lobe has the primary olfactory cortex. These signals do not go through the thalamus first. Right, this is one of the examples. The olfactory signals can reach the cerebral cortex directly without going to the thalamus first. This is not true of any other sense but olfaction, but our sense of smell. So the olfactory receptors all form cranial nerve 1. So the cranial nerve 1 is the olfactory nerve, which carry them to the olfactory bulb. The nerve impulses are passed to neurons of the olfactory tract, which then go to the olfactory centers of the cerebrum, specifically in the temporal lobe, where the scent is perceived. So this is an overview of the projection pathways of the brain. So after we go from the olfactory bulb, this is the olfactory bulb. From the olfactory bulb, you go through the olfactory tracts to the primary olfactory cortex of the temporal lobe. From there, signals travel to several secondary destinations into the cerebrum and the brainstem. Two of these important destinations are in the insula, which is that hidden lobe of the cerebral cortex, and also to the orbitofrontal cortex. And the orbitofrontal cortex is where we identify what the odor is, and it also helps receive input from our tongue, from taste. So the orbitofrontal cortex can combine taste and smell to give us an overall perception of flavor. In addition to the orbitofrontal cortex and the insula, the primary olfactory cortex also sends messages to the limbic system, like the amygdala and hypothalamus. Right? And this is where some of the scents we are exposed to evoke emotional responses. So certain smells might be very pleasing or might be very unpleasant. It can also make you salivate, make you want more of something. Right? So these involve emotions and memory. So that's where the amygdala comes in, where we have emotional regulation associated with smells. Right, and like I mentioned, olfactory signals reach the cortex directly without passing through the thalamus. This is an exception to the rule. So it all started in the olfactory mucosa, in the nasal passage. These olfactory nerves, cranial nerves 1, receive signals from the sensory neurons of the olfactory receptors, and these feed into the olfactory bulb, which send the message toward the olfactory tract. Those go on the underside of the frontal lobes, where they go to the primary olfactory cortex. And from there, we know that there are secondary destinations like the insula and the orbitofrontal cortex. So that was an overview of smell. And the key point to remember is that food is made up of many different odorants, or a smell of perfume is made up of many different odorants. So it's many different receptors are being activated at once, and it's the combination of different receptors activated that give a message to the brain of what you're smelling, or what that particular scent is. So now we'll talk about taste. So this is a good time to pause before we go on. So now we'll talk about gustation. And the types of receptors responsible for gustatory responses are chemoreceptors. And we have chemoreceptors located on our taste buds in the tongue. And when you look at your tongue, you might notice these bumps. Those are not taste buds. Those are called lingual papillae. Lingual papillae. And we have many different kinds of lingual papillae. We have 4 different kinds of lingual papillae. 1 without taste buds, 3 with taste buds. And only one type of lingual papilla really has a lot of our taste buds on them. So we have about 4,000 taste buds on our tongue that are located in lingual papillae. This is an actual taste bud over here. This is one taste bud, this little circle. If we were to zoom in, let's look at the tongue over here. This is a lingual papilla, this whole little bump. And if we were to zoom in, you can see these little circles, each of those are taste buds. So each lingual papilla, each bump on your tongue, has many of these taste buds. Each taste bud could have up to 250 different, each lingual papilla can have up to 250 different taste buds in it. Each taste bud, if you were to zoom in, each taste bud is a group of cells. It could be 50 to 150 different cells that are grouped together in a bud arrangement. And these types of cells include chemoreceptors and other supporting epithelial cells. So a taste bud is multiple kinds of cells that are grouped together in a bud. There's a taste pore on the epithelial surface of the tongue. This is a taste pore, and that's a small pit where certain tastants, that's a chemical stimulus, so a tastant can go through the taste pore and interact with a gustatory hair that extends from the chemoreceptor through the taste pore. So we have these gustatory hairs, very similar to the olfactory cells, like I have those little cilia. In this case, these are microvilli called taste hairs, and those are the receptor surfaces for tastants. So we said the taste hairs go through the taste pores, and each of these taste pores leads to a taste receptor. From the taste receptor, we have sensory neurons that are attached on the opposite side. So we have a gustatory hair on one side, then we have the taste receptor, and then we have the sensory neuron axon on the other side. So let's zoom in one more time. Here we have the taste pore. We have these little gustatory hairs coming through the taste pore. And in blue, we have these supporting epithelial cells. And in purple, we have taste receptors. And together, those make up a taste bud. And when a tastant interacts with a gustatory hair through the taste pore, these neurons will send nervous signals to the brain saying, okay, you just tasted something. Let's take a step back. And in order to activate a taste cell, a chemical must be dissolved in a liquid first. So that's where saliva comes in. And we have 5 confirmed basic tastes. Each has an evolutionary function. And I will note that there is an old concept of a taste map, and that's proven to not be correct. So we do detect 5 different taste sensations primarily. But all of these are detected throughout the tongue. Right? Receptors for each taste are scattered across the tongue. And we have different receptors for each of these different types of taste. So salty. So our salty receptors detect metal ions like sodium. So salt is sodium chloride. So when we have sodium chloride on our tongue, certain receptors will detect that sodium and send a signal to the brain that, okay, I just had sodium. This is salt. And this is very important for our physiology because we need to have the right electrolyte balance and the right blood pressure, right? So blood pressure is tied to the right balance of sodium in our blood. So we need to crave salt. Animals do crave salt and look out for salt so we can have the right type of electrolyte balance. Sweet receptors are activated by different sugar tastants. So sugary tastants. There are different types of receptors that can detect sweet molecules. They belong to a group of receptors called HT23 receptors. They're a type of G protein coupled receptors. And you should think to yourself, why do you think we need sweet receptors? What's the benefit? Why do you think our ancestors would have benefited from having special receptors that detect sweet molecules? So sweet is associated with foods of high caloric value. So this means that way back in the savannah, if you had somebody who can crave and detect sweet molecules, that means they'd be able to pick up the ripest fruits and get the most sugar, the most energy from foods. So this is called gorging theory too. That there's an idea that we had a craving for sweet foods so we can maximize our intake of calories and get the most amount of energy, especially when we didn't know where our next meal would come from. So we detect sweet foods and we learn that it's a positive thing so we can get as many calories in and get as much energy as possible. However, today we're not in the savannah, and this becomes a little counterproductive because we still crave sweet things even when we have a lot of energy. So we're tempted to go get chocolate ice cream from the freezer because we're craving sweet things, but we're not in the savannah, so we're not desperate for energy. So our brain hasn't caught on yet. We're still tricked into wanting sweet things. Umami is the meaty taste that's associated with sometimes like soy sauce or meats, or tomato has a lot of umami. So it's really produced by amino acids. And that's like a mushroomy, some kind of like Parmesan cheese has a lot of umami. And this is thought to motivate protein intake, since this is the taste of certain amino acids. So it's inspiring us to eat more protein. We have sour receptors that detect acids, such as in citrus fruit. And these can help us know if something is spoiled or not. Something tastes sour when it's spoiled. And similarly, bitter receptors help us know if something is a poison or something is spoiled. So some plants use bitter compounds in their flower to deter animals and humans from eating their fruits. So when we perceive flavor, it's due to the activation of several different taste receptor types. So when we eat ice cream, it's not really only the sweet receptors. We're also tasting some of the sourness from the fruit, some of the saltiness maybe. If it's a chocolate ice cream especially, you have sweet, you have salty, you have bitter. You might have some umami, even some meatiness is associated with some, like a dark chocolate, some sourness. So flavor is quite complex. And as we know, the olfactory system is also heavily tied with flavor. So flavor is a combined perception of olfaction and gustation. Also, the food texture and the temperature of the food and the appearance of the food plays a role. And we have certain taste receptors, certain lingual papillae that are responsible for just detecting mouthfeel and texture of food and not even the taste. So our tongue is responsible for taste, for not only taste, but for texture. And just like in olfactory cells, these chemical molecules are sending action potentials. So we have to think about it. How do we transduce chemical energy or chemicals into an action potential? You can either depolarize the cell directly. So certain acids and metal ions, like sodium, can actually go directly into cells, depolarize them, and cause action potentials that go to the brain. So like H+ is in acids, right? So like citrus fruits. So when you eat a citrus, those protons, those acidic molecules, can directly depolarize cells and tell the brain that you're eating something sour. Other molecules, like sweet molecules, bind to their certain receptor, which causes a second messenger system inside the cell to activate a neuron. So sugars and some bitter compounds bind to receptors that activate G proteins and cyclic AMP, which can then depolarize the cell and send a message to the brain via sending neurotransmitters. So what happens is, at the base of taste cells, neurotransmitters are released, as I'll show you here. So these taste cells release neurotransmitters that get sent to the facial nerve and the glossopharyngeal nerve. So the taste buds send their information to the two cranial nerves. Those all go to the medulla in the brainstem. So step 1, taste buds are activated, so they get depolarized. We said either directly or by second messengers. That causes the release of neurotransmitters that will allow either the propagation or the termination of nerve signals through the cranial nerves. Specifically, we have cranial nerve 7 is the facial nerve, and cranial nerve 9 is the glossopharyngeal nerve. Those are both directly hooking up the taste buds to the brainstem. So all fibers reach the medulla from the cranial nerves. Specifically, it's called the solitary nucleus. So all the taste fibers project the medulla in the solitary nucleus. And this is where second order neurons project to the thalamus. And in the thalamus, so this is the second order neuron, going from the solitary nucleus in the medulla to the thalamus. And from the thalamus, it's going to relay the signal to the gustatory cortex of the insula. And that's where we can go, and that's where we perceive taste, is in the insula. That's where and then from there, those process signals get sent to the orbitofrontal cortex, where they get perceived, they get integrated with the olfactory signals and also our visual signals, where we understand what we're tasting. So again, this is a three-step pathway. We start from taste buds. We have taste receptors in our taste buds that each respond to different tastants. When a tastant finds its taste receptor in a taste bud, it gets depolarized. That depolarization leads to neurotransmitters being sent to these cranial nerves that first get sent to the medulla. From the medulla, they go to the thalamus. From the thalamus to the gustatory cortex and the insula. And from there to the orbitofrontal cortex. So you can watch that video as a good overview. Right, so this is showing the odorant, the tastants are interacting with the taste hairs and the taste pore, and that's sending action potentials down. I'm going to go skip some of these disorders. And this is a good time to pause before we talk about the last sense. We're not going to discuss hearing and balance in this course. We're going to finish off with vision. So now we will start our discussion on vision. So take a good look at this picture and try not to move your eyes. So try to fixate on your computer screen and just look at the flag and count to 30. I'll wait. So keep fixating. Your eyes should not be moving. And they should still be fixated. And what did you just see? You should have seen a brief flash of an image of the flag, but in the red, white, and blue colors. And you can try that illusion again. And this was brought up in the crash course. The link is right here. And we will be able to explain the answer a little more clearly after we talk about vision. So I'd like you to stop here and watch this video that's embedded in the PowerPoint that discusses how the eye works on a very basic level. So like an overview of how vision works, because vision is very hard to understand. And this is a 3-minute summary video that's really well made. So that's something to do right now before we get any further. So what is vision? Right. Vision is the perception of objects in the environment by means of light they emit or reflect. And let's just take a time to define what light is. Light is visible electromagnetic radiation from the sun. So the sun has a spectrum. There's an electromagnetic spectrum. So there's all of these forms of energy that reach planet Earth from the sun. But most of the solar radiation of shorter and longer wavelengths is filtered out by ozone and other gases in the environment. So the radiation that reaches the surface of the Earth falls within a very narrow range, and even a more narrow part of that is visible to us. So human vision is limited to wavelengths of light from 400 to 700 nanometers. So this is the whole spectrum. We have on one end, we have the very high frequency gamma rays. And then the other end, we have very low frequency, long wavelength radio waves. And radio waves and microwaves and infrared rays are all invisible to us. Microwaves can heat up water molecules in food, for example. Radio waves can help transmit sound energy. Infrared can be used for imaging techniques, and you can heat something with IR. And we could use UV light and X-rays, for example. But these are damaging. These would be outside of the visible spectrum of light, and X-rays are not visible to our eyes, and in fact damage DNA. What light does within the narrow spectrum of 400 to 700 nanometers, what the light does, it causes a photochemical reaction in the retina cells and in the retina of the eye to produce a nerve signal that's sent to the brain. So light is the stimulus in this case, and these sense receptors are in the retina of the eyeball. And of course, the optic nerve, which is a cranial nerve, links the retina to the brain. And just to give you more specific examples, UV light, like I mentioned, has way too much energy, and that's why it destroys macromolecules. So the reason why it destroys DNA, for example, it can break a DNA double helix, is because it has way too high frequency, way too much energy. IR, on the other hand, has too little energy to cause reactions. So it wouldn't really be functional to cause a photochemical reaction to produce a nerve signal. So we have this very narrow range of visible light that comes from the sun, and we know the spectrum is red, orange, yellow, green, blue, indigo, violet. Those are the colors of the rainbow, so to speak. So eyes are the organs of vision. I think that was something you've known for quite a while. They're located within the orbits of the skull, and there are 7 skull bones that protect each eye. So the skull bones themselves are, I think, pretty obvious. The frontal bone, I mean, some are not obvious actually. The frontal bone in front, and we have the maxillary bone and the zygomatic bone, right, make the anterior outer boundary of the orbit. And in the back, we have the sphenoid and the ethmoid and the lacrimal bones, as well as a little bit of the palatine bone, which you don't see here. And in addition to the bone protecting the eye, we have connective tissue that provides support and protective cushioning for the eyeball. And the eye contains more than 70% of the body's sensory neurons. So of all of our sensory perceptive neurons, they're mostly located in the eyes. And the eye, our vision, is supposed to be our principal sense, our most refined sense as humans, is our stereoscopic vision and our color vision, meaning our ability to distinguish between depth, like our depth perception, and our ability to distinguish between colors. Other organisms can detect even more colors than us, so I don't want to put humans on a pedestal, but I will say that of all the senses, the one that we do best is probably vision. Outside of the eye, we have some accessory anatomy to talk about. So the eyelids protect the anterior surface of the eye, and they help us blink. And what blinking does is it helps spread tears and mucus. So as we blink, the secretions from the lacrimal gland, which are tears, are able to be spread as we blink. And the conjunctiva is this very thin membrane. It's it's transparent, and it lines the internal eyelid surface and the anterior surface of the eye. So the eyelid surface and the anterior surface of the eye, and this is what produces mucus. So this has goblet cells to produce mucus to lubricate the eye. And it also has many blood vessels and nociceptors. So the conjunctiva is very susceptible to pain. So this is why sticking a needle in your eye might be painful, not the best idea. Outside of the eye, we have eyelashes that help keep anything floating in the air from getting into the eye. It also has minimal protection from excessive light. The eye also can help trigger the blink reflex. So the eyelashes can help, if like a particle hits the eyelash, then you're gonna blink right away. The eyebrows shield the eye from any light that's directly overhead. So the eyelashes and the eyebrows both have that same kind of function, and they have a slight shielding effect They help minimize excess light from getting into the eye. But eyebrows have the additional effect of diverting sweat. So if you perspire from your forehead, if you didn't have an eyebrow, it would go right into your eye. So your eyebrow and your eyelash kind of they have very similar functions when you think about it. They both keep things out of the eye, help keep light out of the eye, and help, while the eyelashes help trigger the blink reflex. But I think they pretty much do the same thing and are not as needed in humans as other organs, right? So we don't really need eyebrows or eyelashes. You would be able to be just fine, survival. So like we would say, it doesn't really add to fitness at all. So that can be something that was kind of just passed along through evolution. Okay, so here's just some other images. This shows the lacrimal gland that produces tears. And as we blink, the tears move medially and they get collected in the nasolacrimal duct. So again, so tears are secreted by the lacrimal gland. This is the lacrimal gland. And ducts, there's excretory ducts, carry tears to the eye surface. And the purpose of tears is to lubricate and wash the eye of any debris, but to also deliver oxygen, nutrients, and enzymes. So tears are important for nutrition and protection. And there's an important enzyme called lysozyme that I'd like you to know about. So lysozyme is an enzyme that's also found in mucus and egg whites and lots of other things. But in our tears, we have a lot of lysozyme, and it's like a scissor that breaks up bacterial cell walls. So if we get bacteria in our eye, we have lysozyme that chops up the bacteria so they can't infect us or can't get inside of our eye or our brain. And again, so tears are spread medially and inferiorly every time we blink. So that's the purpose of the eyelid. And the nasolacrimal duct will carry the tears into the nasal cavity. And when we cry, we oftentimes have like a runny nose. You need a tissue. And this is explained by the nasolacrimal duct. Right? When you cry, you have excess tear secretions flowing into your nasal cavity. So you're going to have to use a tissue, blow your nose. So there are muscles that control the eye. There are 6 extrinsic muscles of the eyeball. They all originate on the back of the orbit, and they insert on the eyeball itself. They're called the rectus. We have 4 rectus muscles. We have a superior rectus muscle. We have a lateral rectus muscle. We have an inferior rectus muscle and a medial rectus muscle. So we have superior, inferior, medial, lateral rectus muscles. And those do exactly what the name suggests. So superior moves the eye up, inferior down. Medial will move it to the left, lateral to the right, depending on the eye. And you're going to have the other 2 muscles are called the obliques. Superior oblique and the inferior oblique, but they all work together to function coordinated. They all enable very fine movements of the eye together. And the oculomotor nerve is one of the nerves that control this. So the brain works through the cranial nerves to control the eyeball's functioning and the coordinated movements for vision. So here's a view of the rectus muscles. Here's the inferior oblique, superior rectus, inferior rectus, the lateral rectus, and the medial rectus is on the other side. So let's talk about the eye itself. And the eye is composed of 3 layers. The wall of the eye is composed of 3 layers, and it's hollow inside. And it's about 1 inch in diameter. So think about the eye as a hollow sphere with a wall made of 3 layers. There's the fibrous layer, the vascular layer, and the neural layer. Inside, we have the spaces are filled with fluids that help support the eye's structure and shape. So this is a good picture that we could keep on referring to in discussing the anatomy of the eye. So already in this picture, you can see a nice cross-section that shows the 3 layers. You have the sclera, the choroid, and the retina. So these are the 3 layers of the wall. And then this whole chamber over here is filled with, I'm getting ahead of myself, but these 2 chambers over here are filled with fluids. So let's talk about the fibrous layer first, which is the outermost portion of the eye. And the fibrous layer itself has 2 parts. So this is the outermost layer of the eye, the most superficial part of the eye. And this has 2 parts, the fibrous layer. The sclera is the white portion of the eye. So the whites of the eye are called a sclera. And it's very, very, very tough. And I wish we had an opportunity to do a dissection because you would see how tough the sclera is when trying to puncture it. It is very hard. It is very firm, very fibrous. In addition, the other portion of the fibrous layer is called the sclera. And the sclera is I'm getting ahead of myself. The other portion is called the cornea. But before I talk about the cornea, I'm going to talk about what the purpose of the sclera is. The sclera is the major protective organ of the eye. If you get something like a stone in your eye, you're not going to rupture your eye. You're not going to be able to affect the optic nerve, at least, because you have so much protection from the sclera. It's so fibrous and multi-layered. And we said that the anterior portion is covered with conjunctiva, which produces mucus. So the whole purpose of the sclera is to protect the eye and the optic nerve, what's behind it, right? We don't want to harm this neural layer, the retina, because it's directly connected to the optic nerve, which is linked to the brain. So we need this very tough fibrous protection. And the cornea is the other part of the fibrous layer that is it's a clear window. And the purpose of the cornea is to bend light rays as they pass through it, so they hit the right part of the retina at the right time. So again, to summarize, the eye wall has 3 layers. The outermost layer is called the fibrous layer, right? These layers are also called tunics. So the tunic is a layer. The outermost tunic is the fibrous layer that has the sclera and the cornea. The sclera is white. We have a lot of protection. It is so dense, it's dense connective tissue. So it is great protection against any damage to the nervous system behind it. And then the cornea, we said, is like a window. It's a clear window that helps bend light rays to hit the retina in the right way. The cornea is clear and it lacks blood vessels and nerves. So you can't feel anything if your cornea is scratched. I think. Actually, it should be in theory. I've never got my cornea, but if there's no nerves, it should not hurt. However, the conjunctiva, it would be pretty hard to avoid the conjunctiva because that is very rich with nociceptors. So this is a good diagram that shows cornea and then sclera together making the fibrous tunic or the fibrous layer. Now we can talk about the middle layer called the vascular layer or the Tunica Vasculosa. And this includes 3 parts. It includes the choroid, the ciliary body, and the iris. So the choroid is located everywhere except just the anterior portion of the eye. So remember, we're talking about a layer that goes all the way around. So there's choroid is shown over here. This choroid is this pink stuff. Well, actually, it's really not a good picture of that. The choroid is all the blood vessels that nourish the eye. So you can't, I want to see if I have a better again, really important to see in a dissection. But you can imagine the pink part is where the choroid would be. And it's really rich with blood vessels, so that's what nourishes the eye the most. So think about vascular layer, blood vessels. Blood vessels nourish the eye with oxygen, but it's also filled with melanin. So if you were to do the dissection, you would see that the choroid is kind of black in the sheep eye. And the black pigment helps any backscattering of light, so it can focus on the retina. And like I think of the example of like a movie theater, like you want to make it dark in a movie theater so you can focus on the screen. So that's what the melanin in the choroid kind of does. So you can focus the light on the retina, which is the screen. And what's shown here is the ciliary body, which is a muscle. So the ciliary body has ciliary muscles that surround the lens. And the ciliary body, so it's an extension of the choroid in the anterior portion. So the choroid is all over here, all around here. And then here we have the ciliary body. And the ciliary body helps support the iris, which we'll talk about. And it helps keep the lens in place. So there are things called suspensory ligaments. These are these little strings here. These suspensory ligaments reach out from the ciliary body and hold on to the lens. So that's a very important function of the ciliary body. The ciliary body also secretes aqueous humor, which we'll talk about in a second. It's that fluid of the eye. So it secretes the aqueous humor, which is really very important for vision as well as the physiology of the eyeball. So that's the ciliary body, suspensory ligaments, and I could talk about the iris. The iris is still part of the vascular layer, and that's the color portion of the eye. And it's basically a diaphragm. It's an adjustable diaphragm that can open and close to control the diameter of the pupil. So it controls the amount of light that can enter the eye by changing the size of the pupil, whether it's dilated or constricted. So that's the iris. And the pupil itself is not, it's an opening. So the pupil is not a physical structure. The pupil is the opening in the center of the iris that allows light to pass through. When you shine bright light on the eye, the pupil will be constricted because the iris is going to try to control the amount of light that gets in. You don't want to bombard your retina with all the stimulation. So your iris is going to constrict the pupil in bright light, but in dim light, you need to be able to see. So your iris will control the amount of light, will allow more light in by controlling the size of the pupil. So that was the middle layer, the vascular layer. Finally, we have the inner layer, also called the tunica interna or the retina. So we'll talk about that in a bit, but that's the star of the show, right? The retina and the optic nerve make up the neural layer, and that's the movie screen that's going to be communicated to the brain. The lens is an interesting portion of the eye. So the lens can change shape, but it's very firm. When it gets preserved, so in a dissection, the lens actually gets more like a marble. So it gets very firm, but it's a little bouncy and it's transparent. So they're made up of multiple fibers that are flattened together. There are all these transparent cells that are flattened together, and they form this disc of sorts. This is the lens. This is an electron micrograph of the lens. And like I mentioned before, they're connected to the ciliary muscle by suspensory ligaments. So this is a better image. This is a better way to see the ciliary muscle. So the ciliary muscle goes all the way around the lens, and what connects them are the suspensory ligaments. And this is important because the lens has to be able to change its shape depending on what we're looking at. This is called accommodation. The idea of the lens changing its shape is called accommodation. So we need to be able to accommodate, we have to be able to use accommodation depending on what we're looking at, so light can be refracted perfectly on the retina. So what vision is doing, right? Light is being bent so it gets focused precisely on the retina, specifically in a specific area called where that's rich with the photoreceptors that we'll talk about in a bit. So in the case of distance vision, actually I'll get ahead of myself a little bit. So we need to bend the light. That's the purpose of getting. We need to see something. We need to bend that light that gets refracted from the image. We have to bend it back to our retina so it can then be communicated with our brain. So what's going to be able to refract that light? Right? How are we going to be able to take what's out here and make sure that it hits right over here? It's almost like using a series of mirrors in a way to kind of refract a beam of light in just the right position. So the cornea does a lot of refraction. And you can read a little bit more about what the refractive index is. So light travels through air at a certain refractive index very easily. But any other material bends light. Like water bends light. The light does not travel directly through water. So the cornea is pretty aqueous, and the cornea does a lot of the refraction. So the light gets bent and focused on the retina. And eye optics cause the image to be inverted on the retina, just like a mirror. So even though the A is upright, it gets focused as an inverted A on the optic nerve. And what happens is the cerebral cortex corrects this inversion. So it's an illusion, really. So we're seeing them in their correct orientation. And this is illustrated in a microscope, too. Like when you look through a microscope, you're really looking at an inverted image that's being corrected by a mirror in the lens. So accommodation of the lens helps to focus light on the retina, so we can adjust the shape of the lens depending if what we're looking at is far away, very close, or kind of a perfect distance. So for distant vision, we call that emetropia. Emetropia is distant vision. And emetropia is a state where the eyes are completely relaxed and they're focused on an object that's more than 20 feet away. So at that point, the light rays coming from the object are basically parallel, and they're focusing on the retina very effortlessly. But if we go and shift our eyes to something closer, then we need to be able to focus on something specifically. We need to be able to shift, we need to be able to accommodate the lens, so what we're looking at hits the retina appropriately. It has to still hit the retina. So we have to bend that light so it hits the retina in the right location. So in distant vision, emetropia, the ciliary muscles are relaxed, and the lens is pretty flat. So this is up here. So distance vision, emetropia, the lens is pretty flat. We don't need to do a lot of bending. And when that happens, the ciliary muscles over here are relaxed, so they hold on very tightly to these suspensory ligaments. So when the lens is flat, try to pick so it's taking up more space. So these are very tight strings when the lens is flat. They're pulling on the lens. The ciliary muscles are relaxed. What happens is the ciliary muscles push. They don't pull. So in near vision, the ciliary muscles contract and they push, they move the ciliary body toward the lens to thicken the lens. We're thickening the lens so it could then bend more light to see something that's closer to us. And when that happens, then the tension on the suspensory ligaments is low. They become more slack. And again, try to visualize this. The ciliary body is relaxed over here, and that means that they're held on very tight to the lens, to the suspensory ligaments. There's a lot of tension. And when they push down, when these guys push down, then there's not going to be as much tension on the suspensory ligaments. They're going to be floppy. And that will push the lens in the middle to thicken it. And that could make it able to bend more light. So what you're looking at very close to you can be refracted appropriately on the retina. I know it's a little confusing. I hope I didn't butcher that too badly. This is a great image. I should have just shown this one. So here you see in emetropia, the lens is flat. And you can see that the suspensory ligaments are very tense because the ciliary muscles relax. But when we need to accommodate for near vision, we need to thicken the lens. So we need to refract more light on the retina. So what happens is we need to contract the ciliary muscles to push down on the lens to thicken the lens. And in response, the suspensory ligaments will be more relaxed, right? They're not pulled as in here. As the lens thickens, those become more relaxed. And that's because of the ciliary muscles are contracting. So there are chambers and compartments of the eye. We'll talk about compartments first. So the eye has an anterior compartment and a posterior compartment. We'll talk about the posterior compartment first. It contains vitreous humor, which is that jelly-like substance that's between the lens and the retina. And the purpose of this vitreous humor is to press the retina against the eye wall. Just like developing film, you want to have a flat layer of film. You want to press down on the film perfectly. You can't have any air bubbles. So this jelly-like material inside the vitreous humor keeps a nice seal. It keeps this retina flat against the eye wall. Right? Specifically, it's flat against the choroid that's in the middle, right? That was the vascular layer we spoke about. So that's the posterior chamber, filled with vitreous humor, keeps the eye shape, gives it structure, keeps it round. The anterior compartment is broken up into two chambers. So this is where it gets a little tricky. So there's an anterior compartment and a posterior compartment, but the anterior compartment has two different chambers to it. There's the anterior chamber, and that's simply the space between the cornea of the fibrous layer and the iris of the vascular layer. That's the anterior chamber. We'll talk about that in a second. The posterior chamber is the space between the iris and the lens. So in this cross section, you can see this is the posterior chamber. Here you can see this is between the iris and the lens. This is the posterior chamber. Between the iris and the cornea is the anterior chamber. Both chambers of the anterior compartment are filled with aqueous humor. So you can remember A for aqueous and anterior. And aqueous humor is a little different than the vitreous humor. The aqueous humor helps regulate the internal pressure within the eye and also helps keep the cornea its proper shape. We want to make sure that the cornea is its proper shape so it can refract light on the retina. The aqueous humor also provides nourishment to the cornea and the lens, just like tears do. So tears are first filtered out of capillaries. So that comes from blood. So the ciliary body capillaries filter out the blood to make tears that are first secreted into the posterior chamber, which is between the lens and the iris. And then from the posterior chamber, it goes into the anterior chamber. At the scleral sinus, it is reabsorbed into the blood vessels, and that's where the humor returns to the blood. So this is, again, a liquid that's for the eye. It's just for the anterior compartment of the eye. And this aqueous humor gets filtered from the ciliary body, and it flows from the posterior chamber to the anterior chamber into the scleral sinus, where it goes back into the blood. So at this point, we spoke about the sclera, we spoke about the choroid, that's in red, we spoke about the retina a little bit, we'll talk more about that. We spoke about the vitreous humor, we spoke about the ciliary body, the suspensory ligaments, the lens, the iris, the cornea, the pupil, the anterior chamber, posterior chamber. So at this point, you might want to review everything again if you don't understand everything on this slide. Now we'll go to the third and deepest layer, called the neural layer. And the neural layer is what includes the retina and the optic nerve. So the conversion of light energy into action potentials occurs in the retina. And it's technically part of the brain. It is attached to the eye at the optic disc. So the optic disc over here is the exit of the optic nerve. So the optic disc is where the retina is attached to the optic nerve, which is right over here. And the retina, like I mentioned before, is pressed against the rear of the eyeball by the vitreous humor. We want a flat seal of the retina against the eyeball. If you get a detached retina from like an injury, that could cause blurry areas of vision and can cause blindness. And again, think about an example of like when you're trying to press, if you're developing film, you have to press the film down on something, and it has to be a perfect seal, or else you're going to get an air bubble. Or even think of like a screen protector. You have to put it flat against the screen. Otherwise, you might have little blurry areas. You might have patches on your phone. You won't be able to see through it clearly. And if you really have a detached retina, then you might be blind. And there's corrective surgeries for that if you do get a detached retina. So, how does an image form on the retina? We said that light has to pass through the lens to form a tiny inverted image on the retina. And we said that the amount of light that passes through the lens is determined by the pupil diameter. And the pupil diameter is controlled by the iris. There are two sets of contractile muscles that control the iris. There is the pupillary constrictor, and this is smooth muscle that encloses the pupil. And parasympathetic stimulation will close the pupil. It will narrow it like this, like almost like a wheel. So the pupil constricts, the circular pupillary constrictor muscles narrow. It will narrow the pupil. And that's in response to parasympathetic stimulation. We don't need to see as much. We don't need to maximize as much light getting through. We have other muscles outside that are called the pupillary dilator muscles. And those look like spokes of a wheel as opposed to the tire of a wheel. So it's like an inverted tire almost. You have the constrictors around the pupil directly, and then the dilators are outside of the constrictors. And in response to sympathetic stimulation, these dilators will contract. And what that does, it pulls on the iris. It pulls it outward to increase the diameter. So we're going to get as much light as possible. So in bright light, our pupillary constrictors will contract. In dim light, our pupillary dilators will contract. So when light intensity changes, we're going to have different contractions of these pupillary muscles. When our gaze shifts between distant and far, we also need to accommodate different amounts of light so the image can focus on the retina. And that's what's shown over here. I'm not going to talk too much about this, but we oftentimes have some kind of defect in vision. So some kind of defect in image formation could be corrected, right? You could go now very easily and you can get contacts or glasses. So emetropia, normally we have an image that is perfectly refracted onto the retina. So what this is showing is as light enters through the cornea, the lens refracts it. The lens bends the light so it hits the retina exactly where it needs to be. However, if you are farsighted, that means the eyeball is too short. So what happens is that the eyeball is too short, the light gets bent behind the retina. So that's, again, hyperopia is farsightedness. So that's when the eyeball is too short, right? The retina is in front of where the light is hitting, or the focal plane. So the focal plane is how we define where the light is going to hit. So the focal plane is behind the retina if you're farsighted, because your eyeball is too short. If the eyeball were longer, then the light would hit the focal plane. The focal plane would be at the retina. So a correction for hyperopia is to use a convex lens. And the convex lens will cause the light rays to converge slightly before, right? So we're seeing some convergence before it gets to the lens. So therefore, even though the eyeball is a little short, that convex lens bends the light just a little before it gets to the lens so it can hit the focal plane. If your eyeball is too long, you can imagine that the light rays will then come into focus before they reach the retina, and we would need to correct that with a concave lens. So a concave lens would cause the light rays to diverge slightly. So they're kind of get spread out more. So look how they're narrow over here. The concave lens causes them to diverge a little bit. So when they get to the lens, they'll be refracted properly onto the retina. So even though the eyeball is too long, a concave lens can help refract the light just where it needs to be. And that's all you need to know about lens formation, lenses and image formation. So this is a good time to stop, because this is where it gets a little tricky in terms of the cellular level of cells interacting with each other. So you might want to pause here and make sure you understand everything anatomically we spoke about so far. Okay, so the retina is what converts light energy into action potentials. And a little fun fact is the retina is the only part of the brain that you can see without dissection. So using an ophthalmoscope, you can actually see the retina, which is part of the brain. And the retina has two types of photoreceptors. And again, photoreceptors, remember, means light receptors. We have rods and cones. So these are two special cells of the retina. You can see them over here, rods and cones. These are called photoreceptors because they're activated by light. Rods are used for black and white vision. These are very sensitive, but only to the presence of light. It's like, is there something there or not? So this is really important. So rods are very important, like in darkness when we need to just see if there's like an object attacking us, or or if we need to find something. Like rods are for black and white vision. Like when we just wake up, our rods are sensitive. So they're sensitive is the key word. Cones are for color. So the cones require bright light to function. So that's why usually when we wake up in the middle of the night, it takes some time to get full color vibrancy in our vision. Because we first we only have our rods function. So we kind of see in black and white. The cods are cods. That's if you combine a cone and a rod, you get a cod. But a cone is essential for the resolution of images. So the fine details are due to the cones. And we have 3 different kinds of cones, at least. They kind of branch off into different types from there, but each could detect a different color. Maybe we could have like red or green or blue sensitive cones. And cones and rods together allow us to have a perception that is sensitive and highly resolved. So we can get nice resolution and contrast and sensitivity due to our photoreceptors of the retina. There is an area of the retina that's called the macula lutea, or the macula lutea, which is directly posterior to the lens. So it's directly behind the lens. And it has an area called the fovea centralis. And that is where the cones are most concentrated. So you can remember centralis, the fovea centralis is where the cones are most concentrated. And as you go from the fovea, the cone density decreases. So as you go, and this is, we're looking at like a cross section. We're looking at like a lateral view of the eye over here. So this is the optic disc. The cone density is going to decrease with the distance from the fovea. And the rods are least concentrated at the fovea and increase density as they go outward. So again, we have the opposite relationship. Cones are most concentrated at the fovea, rods are least concentrated at the fovea, and the density of rods increases as you go from the fovea, whereas the cone density decreases as you go from the fovea. And behind the retina, we have that pigmented epithelium, and that contains melanin. And again, that's going to be responsible for preventing backscattering of light. So we had melanin in chromatophores of the vascular layer, but we also have melanin in the pigmented epithelium. And again, that's like in a camera. You want to make sure that it's black on the inside so the film can develop. But you want to make sure that if you're going into like a dark room, it has to be dark. You don't want any outside light interfering with you developing your images. You could examine the rear of the eye, the fundus, with an ophthalmoscope. So that's how you look at the retina. And you can see the macula pretty well. Over here, that's the spot. This is the macula. And you can see that the fovea, which is the pit in the center. And the optic disc is also very easily seen with an ophthalmoscope. And that's where the optic nerve exits the retina. So the optic disc is the location where the optic nerve is right behind. So there we have blood vessels also exiting the retina. And this will give us what's called a blind spot, as I'll show you in a bit. So here we have the neural layer. So we have the retina. Optic disc is where it's connected to the optic nerve. This is where the blind spot would be. So the retina also contains other neurons. So the nerve impulses from the rods and cones over here, our photoreceptors, are then transmitted to bipolar cells. Bipolar cells. And from there, the nerve impulse is transmitted to retinal ganglion cells. It is the axons of these RGCs, or the retinal ganglion cells, that all converge at the optic disc to form the optic nerve. So it's very important to take a second to try to visualize this, because this looks like a complicated image. Let's try to understand. This is we're looking above the eye. This is the back of the eye. Up all the way down here would be the lens. So light is being refracted from the lens into the back of the eye. And it first is going to hit the back of the eye and the pigmented epithelium. So the first cells to receive the message are the photoreceptors, the rods and cones. They then transmit the signals to the bipolar cells, which then go to the ganglion cells. Those all converge at the optic nerve and exit right to the brain. Since there's that part that exits the eye to the brain, that's going to give us an area with no photoreceptors. Right? You can't have any photoreceptors where all the axons are leaving. So that's going to create the blind spot effect. I think I have a slide coming up that will show you the blind spot. We have other cells called horizontal cells. Those form horizontal connections between the photoreceptors and the bipolar cells. So horizontal cells make horizontal connections, and they kind of enhance the perception of contrast and of edge detection. So horizontal cells enhance our visual perception. They help integrate signals. And again, amacrine cells have a similar effect. They also, the amacrine cells specifically, help integrate the message to create better visual perception. So the horizontal cells I'm trying to think of a word. They kind of translate the message. They integrate and translate the message between the rods and cones and the bipolar cells. And these amacrine cells kind of do the same thing between the bipolar cells and the RGCs, the ganglion cells. So we have altogether of the retina, rods and cones are the photoreceptors. We then have horizontal cells, bipolar cells, amacrine cells, and ganglion cells. These are all the cells of the retina. Altogether, all these cells enhance perception of contrast, edge of objects, moving objects, and changes in light intensity. So here's a nice picture. This is the front of the eye. This is the vitreous body. This is the retina. This whole from here to here is the retina. And within the retina, here's our ganglion cells. Here's some bipolar cells. You can't see the horizontal, they're not stained, the horizontal cells. The rods and cone nuclei are over here. These are the outer segments of the rods and cones. And this is that melanin-rich pigmented epithelium. So that marks the end of the neural layer. Then this is the vascular layer, choroid. You can see the blood vessels. And then you have sclera, which is, you can see some, this is all collagen, dense connective tissue that is very tough and fibrous. So the optic disc, like I mentioned, so that's this area right over here. You can't have any photoreceptors here. So the optic disc is where all the blood vessels enter and exit. So we need to have capillaries that are continuous so we can nourish the eye all throughout the eyeball. So blood vessels enter and exit at the optic disc, and those vessels are continuous with capillaries all throughout the eye and the vascular layer, so they can nourish all of the eye. And again, since there are no receptors here, we call that the blind spot. And you could use the book talks about this illustration of the blind spot. Let me see what page it's on. I think it's on page 608. If you close your right eye and stare at the red dot, keep on moving toward the screen and move your head toward and back from the screen until you see the X disappear. That is your blind spot. What happens is, so again, you should be able to see the X, to see the red dot disappear. And again, because the image of the dot is falling on the blind spot of your left eye, you're not seeing anything there. But then the question is, why do you still see a green line? Right? It's not like you see just white. You still see a green line. And that's because our brain does visual filling. So the brain does this illusion where it fills in it fills in the blind spot with whatever's around it. So we don't walk around looking and having like a spot in our visual field all the time, right? We always have a full image. We can see fully clear. And that's because our brain is kind of playing a trick on us and is filling in the blind spot with whatever's around it. So even though we are really not seeing 100% of what's in front of us, our brain is making us imagine that we are. So the brain ignores unavailable information until saccades, we have these fast eye movements, very fast flickering eye movements, ensure that the same area of the visual field doesn't always project onto the same area of the retina. So we can always use these very fine, fast muscular movements, so we can redirect our gaze and see the entire image and not just get focused on in one visual field. Right? We need to kind of like glance back and forth to kind of get the whole glimpse of what we're looking at. I hope that makes sense. So take a second to watch this video here. Great, so that's a good overview of what we spoke about and what we're about to delve a little bit deeper into. So let's talk about the physiology of vision. And for this, we have to zoom into the photoreceptors. So the rods and cones contain photopigments that can be broken down by light. So that's the first step. We have photoreceptors that have special pigments that can be broken down by light. And when these pigments are broken down by light, the photoreceptors form nerve impulses. So let's just understand that. We have the eye as a sense organ that responds to light stimuli. And we know the photoreceptors are what are in the eye. They have special pigments. Anytime a photon of light hits it, those pigments in the photoreceptor get broken down. And in response, a nerve impulse is sent along to the optic nerve, to the brain. That's the most simple overview I can possibly give. So let's talk about this more. The rods have a pigment called rhodopsin. And when a rod is activated by light, rhodopsin is broken into an opsin protein and a molecule called retinal, which looks a lot like vitamin A. That's why vitamin A is important for your vision, they say. So light causes this photochemical reaction, causes a chemical reaction. It causes rhodopsin to be broken into opsin and retinal. And when opsin is activated, opsin can activate enzymes to form a nerve impulse. So once you have light activating a rhodopsin, the opsin splits from the retinal, and the opsin part can activate enzymes so the nerve impulse can be formed. And to say, okay, this photoreceptor right here was just activated by light. This was just stimulated by light. Cones have a different pigment called photopsin. And the photopsins are different depending on the type of cone. So one type of cone possesses a red sensitive pigment, one has a blue sensitive photopsin, one has a green sensitive photopsin. How we perceive color is a result of the combination of the different kinds of cones and how the different nerve impulses are interpreted and integrated by the cerebrum. So here's an image of, here's a rod, this is a photoreceptor. Within the outer portion, we have these little discs. And within each disc, we have tons of rhodopsin molecules embedded in these discs. So let's zoom in. This is a rhodopsin because you have an opsin protein and this retinal molecule. So this is what we call the photopigment. That's in the photoreceptor. So these discs in the photoreceptor are saturated with these photopigments called rhodopsin. And in response to light, retinal can change its shape. So it goes from this kind of branched shape over here to this shape. It's the transretinal shape. So in response to light, there is a chemical reaction, and the opsin is now going to activate some enzymes. So let's talk about how this whole thing leads to vision. Let's talk about what happens in the absence of light. That's the best way to think about it. In the dark, here is our rod cell, here is our bipolar cell, and here is our ganglion cell. In the dark, our ganglion cells are not firing to the optic nerve. So we're not going to send, the brain is not going to be receiving any message when our eyes are closed. The reason why that is, I'm going to try to go backwards, and maybe this, I'm trying to see if I should, let me take a step back. When glutamate is present, bipolar cells are inhibited, and the retinal ganglion cells are not activated. Glutamate is released in the dark. So when it's dark out, or when you're closing your eyes, these rods steadily release glutamate, which is a neurotransmitter, at the base of their cells. This is the basal end. So when there's no stimulation from light, that means glutamate is released by this rod cell, which means this bipolar cell will be inhibited, which means they're not going to communicate with these retinal ganglion cells, and no action potential will be produced. So take a second to really understand that. So close your eyes right now, and as you're closing your eyes, know that glutamate is being released from your rods to inhibit bipolar cells to not communicate with your ganglion cells. So your brain is not seeing anything. However, when the pigments of rods or cones do absorb light, then the glutamate secretion stops. So the opsin activation, we'll see, stops the glutamate from being secreted. And if no glutamate is secreted, that means the bipolar cells are no longer inhibited. So the bipolar cells can now release a neurotransmitter to the retinal ganglion cells to produce an action potential. This happens because of the opsin enzyme was activated by light. So when light is absorbed by the rhodopsin, we said that retinal changes its shape, right? That's what really happens first. So light hits rhodopsin, the retinal changes its shape, which activates the opsin enzyme. That opsin enzyme inhibits glutamate release from the rod. How it does that is opsin can break down the second messenger cyclic AMP, which is what normally triggers the glutamate from being released. So in its natural state in the dark, there's a lot of cyclic GMP, that's a cousin of cyclic AMP, that is in the rod cell that's making the glutamate being released. So when there's a lot of cyclic GMP, glutamate is being released, which means no message is being sent. However, in response to light, the retinal in the rhodopsin changes its shape. That will activate the opsin enzyme, which will then degrade all of the intracellular cyclic GMP. So no more glutamate will be released. If no more glutamate is released, the bipolar cell will then communicate with the retinal ganglion cells. The ganglion cells are the only retinal cells that actually produce action potentials. They're the last to get the message, really, of the retina. Even though they're the closest to the light source in a way. This is not a good image of... So if you're looking at this tree, even though the retinal ganglion cells is closest to the source you're looking at, it's the last to receive the message in the retina. And these ganglion cells are responding to the bipolar cells as their neurotransmitters increase and decrease. So the ganglion cells respond to the bipolar cells based on their neurotransmitters, and they'll send either very frequent action potentials if you're seeing something very bright, or very few action potentials if you're seeing something that's dimmer. So via the optic nerve, right, that's all being sent through these ganglion cells, these changes will provide signals to the brain of what's going on outside. The photoreceptors that are in the retina have to then transmit all of their impulses along cranial nerve 2, the optic nerve, to the brain. And we know the visual cortex is all the way in the back, right? It's all the way posterior and caudal. So we need to go quite a ways from the retina to the visual cortex. So, and of course, we know we have to make a pit stop along the way in the thalamus. Right? We have to stop at the thalamus, and the thalamus will then relay the message to the primary visual cortex in the occipital lobe. So the photoreceptor nerve impulses are transmitted along cranial nerve 2, the optic nerve, to the brain. And something interesting happens at the optic chiasm. The two optic nerves meet at this chiasm, this crossing. And axons from the medial half of each retina cross over to the opposite side. So let me show you what that means. So here, the medial half of the left retina, right, which is looking at the left visual field, crosses over to the right side of the brain, whereas the medial half of the right retina crosses over to the left side of the brain. So that happens at the optic chiasm, and you can see that in a brain dissection, which unfortunately we won't be able to do, but by watching the videos, you can definitely see the optic chiasm on the underside of the brain. Optic tracts carry the axons from the chiasm to the LGN, which is called the lateral geniculate nucleus, and that's a part of the thalamus. So it goes to the thalamus first, and then the thalamus will relay that information to the occipital lobe, where the primary visual cortex will interpret what the light pattern detected was. So again, it's very, very hard. You have to kind of keep on reviewing vision to really understand how the whole thing works, what the big picture is, pun intended. Right? Think about the world, like your retina is like a camera, and you're looking at the world through pixels. So every pixel can activate a certain photoreceptor in the back of your camera. I hope that makes sense. I'm using my hands right now to try to explain it. But your eyes are like a camera. It's like a moving video camera. And each photoreceptor in the back of your retina is activated by one particle of light. And those particles of light can then send an action potential that will then get sent to a certain place in the thalamus that corresponds to a place in space. This is getting very confusing, but there's like a map in the LGN that corresponds to the visual field. So these neurons get routed to a certain level of the thalamus and then they get transmitted to the primary visual cortex, which can interpret that as an image. The crossing of the medial axons is what allows us to see in 3D. And that's one of the things that makes primates unique, is stereoscopic vision, to allow us to perceive depth. So let's just review the mechanisms of vision. First, light is bent as they pass through the cornea. Depending on how far away the image is and the light conditions of the environment, the iris will control the amount of light passing through the pupil. Right, we spoke about those muscles that constrict in response to parasympathetic or sympathetic stimulation. The ciliary body also helps adjust the lens, so the shape can accommodate and focus the light rays on the retina. As the light hits the retina in the right place, the light is absorbed by the photoreceptors, the rods and cones, which activate bipolar cells by inhibiting the release of glutamate. By inhibiting the release of glutamate, the retinal ganglion cells are then activated and are transmitted to the cranial nerve 2. So the nerve impulses go from the bipolar cells to the retinal ganglion cells, whose axons converge at the optic disc to form the optic nerve. We said that the medial fibers of the optic nerves cross, they decussate, they cross over at the chiasm, and they merge with the lateral axons on that side to form optic tracts. And the optic tracts are what carry the message from the chiasm to the thalamus, specifically the LG end of the thalamus. From there, third order neurons will then transmit the message from the thalamus to the occipital lobe. And then that's where the occipital lobe, which is part of the cerebral cortex, will interpret what we just saw from both sides of the visual field. This is actually a very short image, a very short video on color that I'd like you to watch. Very interesting. How we perceive color in the world around us. Color blindness is a hereditary alteration or a lack of one photopsin. So it could be a hereditary condition. That's most common is red-green color blindness. That's maternally, it's X-linked. So that means that it's more common in men than women because it's a recessive disorder. And red-green color blindness results from either L or M cones, which are two kinds of cones that help distinguish between red and green. So this causes a difficulty between relating the two shades from each other. So if you cannot see a number inside here, that means you have red-green color blindness because you can't distinguish between the red and the green pigments because you don't have the right cones to distinguish between the different stimuli. So the number should say 74. So is it 8% in males? This is actually quite common. So this has been a few years since this controversy. But what do you see here? What color is the dress to you? So to me, it is very clearly white and gold. I know some of you are saying, what? No way. How could it be white and gold? Some of you might see it as black and blue. Maybe that changed it, maybe not, the black background. It has a lot to do with what our brain is subtracting. So it's an illusion. These colors, this is the best image I can get to try to explain it. This image looks white. This apron looks white, and this apron looks blue. But they're really the same color. It just has to do with the background, what's behind it. So our brain sometimes will subtract different colors to try to interpret what one color is. So some of our brains will deduce this as being yellow and white, or like gold and white, whereas some will look at it as being blue and black. So this is like an optical illusion that's still being studied, but it has to do with how our brain tries to subtract color to try to focus on one color. I'm going to talk about, I'm not going to talk about too many disorders. I think I've spoken enough about the eye. It's good to get some familiarity with cataracts and glaucoma. Glaucoma is when there's too much pressure in the eye, intraocular pressure, and that's because aqueous humor isn't properly drained. And if you don't drain your humor, you build up too much pressure, and that can cause the retinal cells to be compressed. And the blood vessels are compressed, so they have no oxygen. And once you have glaucoma, you cannot restore vision. And cataracts are clouding of the lens, and that happens with age a lot of times, but it's also environmental. So diabetes, smoking, drugs can all influence cataracts. But you can get cataract surgery by replacing the natural lens with a plastic lens. So stereoscopic vision is depth perception, right? It's the ability that we can judge how far away something is. And this is something that's unique to primates as far as we know. And the fixation point, right, is the point in space on which the eyes are focused. And we said the fixation point has to be on the retina. But depending on where, if the object is near or distant, that fixation point may be slightly medial or lateral. Because we need to use two different eyes with overlapping visual fields in order to perceive depth. So our eyes are tilted. This is called convergence, right? We can tilt our eyes so we can overlap them. And when we have this overlap, we overlap the visual fields, we can look at the same object from different angles and then perceive depth. This is different from panoramic vision, like a horse or a mouse. They have eyes on the opposite sides of their head, so they can see what's left and right. Like for predators, that's great. But they can't see what's right in front of them in full 3D. And then if you haven't watched this video, watch this again, and maybe you'll gain a different understanding from it. And from here, I'm actually going to call it a class. So these are some more disorders that if you're interested, you can look at this. It's not going to be on the exam, but I can now say that you have all of the information for the last exam that will cover mostly chapters 13, 15, and 16. Anything, it's mostly this chapter. It's mostly chapter 16, with some of chapters 12 and 14. If I had to rank the level of topics, I would say the most will come from chapter 16, followed by chapter 15 and 13, then 14, and then 12. But I'll focus mostly on chapter 16. As always, please email me with any questions. We are not going to go through hearing, but I would love for you to watch the crash course of these so you get an idea of how hearing works. It's quite interesting. Very complex. But now we spoke about taste and touch and sight and smell. So you know 80% or more of our senses. So I hope that was interesting. And this is officially the last lecture. So it's been a pleasure teaching you, even though it's been a very unusual semester. Nobody expected this. And I wish I could have said goodbye to all of you in person. You were a great class. I really mean it, actually. Really great class. And hopefully our paths will cross in the future. And I wish you the best of luck studying for this exam and in all of your future academic endeavors. And always keep in touch. And email me if you have any questions. Take care. And vision is probably the most complex of all the senses, but it is very, very interesting and definitely worth learning about. So the next part we'll talk about vision.